I'm going to show you what to do with these doodle notes for nucleotides. I've opened up the document in Notability, and doodle notes are supposed to be an interactive way for you to be taking notes, supposed to be fun. You can use different colors, color in the titles, color around the outside. Whoops, maybe I wasn't ever a very good doodler. There. Okay, so I'm going to go over what you're going to be doing on each of these pages, and this is going to be your nucleotide doodle notes. So firstly, on this page of nucleotides, I'd like you to color in the atoms, and let's start by coloring in the carbon atoms black. So you're going to color all the carbon atoms in black. The rest you can choose. Nitrogen or oxygen I'm going to put in as blue. Nitrogen I'm going to put in as red. Let's see, can you use white for hydrogens since we already used black? Yeah, that doesn't look like anything. Let's do dark green for the hydrogens. All right, so this practice is going to help you recognize what these compounds look like and the different atoms that are in them. Then you're going to label the parts of the nucleotide. All right, so that's the overview. Now I just want to talk through some of these notes with you. So nucleic acids. Nucleic acids are another type of biomolecule that's important for life. Nucleic acids are made of nucleotides. So nucleotides are the monomers. They have three components. The nucleotides have three components. They can be switched out and switched out and mixed, mixed and matched. So then you're going to finish labeling the parts. You're going to fill in the blanks there. Phosphate groups. Nucleotides use different phosphate groups. ATP, the one on the bottom, can be used to carry energy throughout the cell. So that's where you're identifying the adenosine triphosphate, adenosine diphosphate, and adenosine phosphate. Then up in the pentose monosaccharides. So what are the difference between these two sugars? One is a deoxyribose and one is a ribose. Why don't you circle what the differences are there? And then the nitrogenous bases. So they have two general shapes. The purines have two rings. The purines include adenosine and guanine, or adenine and guanine, A and G. Pyrimidines include cytosine, thymine, and uracil. So the pyrimidines are CT and U in the molecule RNA, and purines have A and G. So label those in the nitrogenous bases as well. So you can see there's lines underneath that to do that. So that's where I'm stopping right now. I will be back with the other pages. All right, so I still have some notes on the back or the bottom of the nucleotide page. One way to help you remember which is the purine, which ones are the nucleotides are the purines, which are the pyrimidines. Pure as gold, so the purines have A and G, and then cut the pyrimidine, or cut the pi, that's a poorly written U. C, U, T are the nucleotides, which are pyrimidines. So pure as gold, cut the pie. Maybe that will help you remember which ones are which. So moving on to our DNA first. So DNA. DNA, where is it? Where is it in the cells? Oh yes, it's in the nucleus. And it is like a giant recipe book. It has the instructions for proteins and all the other functions of the cell. So on this page, what you're going to do is you're going to color in the DNA molecule. And down here are the instructions for colors with that. Let's see, up here. So up here, these are already labeled. The thymine and the adenine and the guanine and the cytosine. Make a note of the hydrogen bonds. So even though in this diagram they look like they're connecting, they're really hydrogen bonded to each other. So hydrogen bonding, intermolecular bonding between two different molecules. All of these would be what kind of bonds in here? These are covalent bonds within the molecules. So here's hydrogen bonds. A and T, we need to know that there's 2 hydrogen bonds that hold those two together, and that guanine and cytosine use 3 hydrogen bonds to hold that in place. And the 2 strands are anti-parallel, meaning one is upside down compared to the other. So on one side it runs one way, and the other side the sequence or the order runs the opposite way. All right, so with the others, let's see what these notes are supposed to say. The 2 strands of DNA form a helix like vertical sides of a ladder. The 2 strands have backbones made up of the phosphate group and the sugar. So you're going to have all along here, this is the backbone. You're going to have the phosphate group and the sugar. The ladder rungs are made up of 2 bases, the nitrogenous bases, 1 base from each strand, 1 from the other. And then these base pairs are always put together with hydrogen bonds, and A is always in a base pair with T, and C is always in a base pair, a hydrogen bond with C and G. Hey everyone, we're now on the last page, the ribonucleic acid page. RNA strands are usually shorter than the long DNA strands and often can carry genetic information like photocopies from DNA to other parts of the cell. Some types of RNA act as ribosome enzymes made of nucleic acid. Enzymes can catalyze or speed up chemical reactions. Many enzymes in the cell are made of protein. There are some ribosomes that can self-replicate or build other RNA molecules. Some can stick to amino acids, helping them speed up reactions that involve proteins. All right, on this page, you are also going to color in ribonucleic acid, single-stranded. It's going to have a different nucleotide base. Instead of thymine, it has uracil. So the structure of RNA can be found, RNA strands can be found in the single-stranded and double-stranded form in the cell. Just like DNA, sugar phosphate groups make up the backbone. The bases stick out of the backbone. RNA strands can base pair with other DNA or RNA strands if they are complementary. What they're thinking in terms of the RNA world hypothesis. Because some RNA can self-replicate and can catalyze reactions, including reactions with proteins, some scientists believe that primitive RNA was one of the first molecules to carry genetic information in very primitive organisms. Many scientists believe self-replicating RNA molecules were used in living things before DNA and proteins. So, kind of like the evolution of nucleic acids, I guess. So you can finish up this. RNA, you probably have heard in the news a lot. The coronavirus is an RNA virus, and a lot of the tests that determine if you've got the virus are RNA based. So that's your doodle notes. Let me know how you like them. We've never done this one before. Have fun.